Welcome back to Unit 2. We're going to do Part B, and that's early life on Earth. And we can see I have a couple different cells up here. We're going to be talking about the difference between prokaryotic and eukaryotic cells in this, the next couple lessons. So one of the first things, we're going to talk about, I'm kind of throwing biological concepts in there, and I'm really kind of giving you just the summary of them so you can kind of have a little bit more understanding as to what is going on during this point of developing the organisms that live on our planet. So we're going to talk about DNA replication, a little bit about cellular respiration, just the beginning, and a little bit about photosynthesis. So about 3.5 billion years ago, we had the first prokaryotic cells, and we've found fossils, right? That we can date, and these are really kind of interesting. We believe these are a bunch of different cells that are bacterial cells or prokaryotic cells that are kind of stuck together with this mucus that they have formed as part of the cell wall. So the first prokaryotic cells, they don't have a nucleus. They don't have other organelles like mitochondria. Okay. They reproduce by binary fission, meaning that they basically have one cell, they elongate. As they elongate, they're going to make a copy of their DNA. So what they're going to do is they're going to unzip the DNA, make a copy, and I'm going to talk about that in a minute, and then they just simply split in the middle, and now we have two identical cells. That is referred to as binary fission. You could also call asexual reproduction. That's another term you can use. We just generally, when we talk about asexual reproduction in bacteria, we have a special name and we call it binary fission. So let's talk about how DNA is replicated. One of the things that we have found is that DNA is a much more stable molecule than RNA. Because the sugars are different, RNA its integrity is not nearly as strong as DNA. In fact, it's important that it does break down. And we'll kind of get into that a little bit. We do not want our DNA to break down, which is what the significance of having that different sugar gave that molecule that advantage. So like I said, DNA is going to use adenine, thymine, guanine, cytosine. They are going to base pair. When I mean base pair, I mean adenine is going to be attracted to and bond with thymine, and guanine will do the same with cytosine. And there's a great Amoeba Sister video also showing this process. So you can see here are double strands. Now our strands actually do run in different directions. So one is running this way, one is running that way. And that's kind of significant in some of the other biological findings, but that's not, I don't want to focus on that. But notice the the complementary base pairing here. A with T, G with C. You can see how they fit together in this model, right? So all we have to do is unzip our DNA, and then we have that strand available to put the base pairs back in. So as soon as we unzip it, each strand is exposed and it can pull in its opposite partner. Those attractions here are called hydrogen bonds, the type of bonds. They're very, very weak, which is important because we need to open and close our DNA over and over and over again. We need to be able to unzip it and then zip it back together. So this process is called semi-conservative replication, and it's called that because, again, we're taking our original strand, we're separating the two strands apart, and then we're making a copy, a complementary copy, so that half the molecule, right? Notice half the molecule is the old strand, and then the other strand has been newly synthesized or made. And we'll keep doing this process over and over and over again. So it's never really destroyed. It just keeps, it's kind of immortal in a way. Now, prokaryotic cells were heterotrophic, meaning that they absorbed or took in nutrients. Okay, they consume their food in order to make energy. They would use this food, primarily sugar, to do the first part of cellular respiration. They're going to convert that sugar into a molecule called ATP, which is what our cells use as the energy currency. So to get the certain processes to work, we need ATP. The first part of cellular respiration is called glycolysis. All organisms on our planet undergo glycolysis. Again, we want rewinding time. This shows those evolutionary relationships. Glycolysis is, we get the name like glycogen, glucose. That's all the basic sugar that we're using here. So glycolysis means to split sugar. So we have our glucose and we're going to split it. Notice we split it in half. So we have 3 carbons. Ooh, that's terrible. We have 3 carbons here, 3 carbons here. We started with 6 carbons. We're simply going to split it apart. There's quite a few steps involved in this, but at the end, we end up with 2 molecules of pyruvate. We also get a little bit of ATP. And we're also going to get this molecule here called NADH, which is also going to be able to be used for energy later. All right. Notice in this process, we did not use oxygen. Okay, so this is a process that is anaerobic. It goes without oxygen. All right. Not a lot of ATP is made with this process, but bacteria cells don't need a lot of energy. Okay, so if we're going to get to multicellular and to organisms that are going to have to do more work, we're going to have a better way of getting energy. But this is going to be the first part, and then we're going to kind of have additions onto it. So anaerobic cellular respiration, because we know that the planet did not have what we call free oxygen. Free oxygen just means O2. We have oxygen. We have the element oxygen on our planet. It's just like tied up in like water or silicon dioxide, which is sand. We didn't just have it by itself. Okay. So there was only one type of, when we talk about this cellular respiration, we're talking about alcoholic fermentation. Okay. So with alcoholic fermentation, it would be able to do that first process of glycolysis and keep it running without oxygen. Again, does not make a whole lot of energy for complex processes. Now, it is very important for the evolution on our planet that there was no free oxygen. Oxygen is very, very corrosive, and it would prevent many of the complex molecules that we need for life to have formed. All right, so we can actually see, we have proof that there was no free oxygen. Okay, when we look down in past sedimentary rock layers, these rock layers where iron was deposited. So we have our iron there. Iron does not rust unless there's oxygen. So we can literally dig down in the layers of rock and we can see where approximately how many million years ago did we start getting free oxygen in the atmosphere. Now, we're talking about life is in the water, right? These anaerobic bacteria, they're in the water. They are taking in nutrients. As they are competing, there was a mutation. Okay, something changed, and some of these early prokaryotes were able to collect energy from the sun, and this is the beginning of photosynthesis. So when we look at the evolution of photosynthesis, we can see how this process goes from just getting. So we have here. Okay, we have our light coming in. We're going to actually use some sulfur in this one, hydrogen sulfide. Some of you guys may know that because if you have like wells, this is one of the chemical processes, and that makes that rotten egg smell is when we have that sulfur, hydrogen sulfide. It's going to pull that hydrogen off and actually put it on carbon dioxide to get sugar. right? And then here we can see the same thing. We're using the hydrogen, um, the hydrogen sulfide in light, and it's just making ATP. So we make ATP. And then here we're going to move over, okay, which is good because I would not want to smell our planet if we keep doing this. We're going to use water instead, and we're going to harvest the electrons off of water to make our sugar. So we can kind of see the progression of this complex process as we get plants that are bigger and more sophisticated. As it became more sophisticated, we are going to start putting out more and more oxygen into its surroundings. So we're in the water. As the water starts to get saturated with oxygen, it will start to leak into the atmosphere. And that's where we're going to start getting atmospheric oxygen. And these are just some of those early bacteria. Well, we used to call them cyanobacteria. And now we're kind of calling them, I don't know, we've split some of the divisions up, but they were commonly called cyanobacteria. And here's the stromatolites. Okay, so stromatolites are what we believe are like one of the biggest photosynthesizers to contribute to the oxygen formation on our planet. Now, if we look at it, they form mats and then they keep growing upwards, and it's kind of like a tree that's adding rings to it. So we can actually go back and see how old some of these structures are. We know that the layer rate is about 1 meter every 2 to 3,000 years. So it's kind of crazy that these organisms we know that we have today, we still have them. They're very famous off the coast of Australia. They are pretty much the same organism that has been here for millions of years. Okay. So before these cells started pumping out oxygen, our Earth's atmosphere had only about 1% free oxygen. After 2 billion years of work, it is now 20%. And then we could see that if we took this little part here, you'd see the cyanobacteria, and then they would just kind of bubble off. The oxygen gas would bubble off of them. So we have looked at, and there were some significant changes on the planet of our planet's environment as we had these different amounts of oxygen. So we can see here how atmospheric oxygen starts off really low. And then here's when we start seeing that the iron in the soil gets oxidized or formed rust. So we noticed this is where we had that rise in oxygen that led to atmospheric oxygen. And then we keep going up. So we have here what we call the Great Oxygenation event. So this is where we're really starting to kick free oxygen up into our atmosphere. Now, the eukaryotic cells. So we still have, we have all these cells in the ocean, the seas. Okay. They are cooking, and as they become, as millions of years progress, they become a little more complex. And so they start to compartmentalize, meaning they start giving jobs for different structures kind of in their cell. For example, a nucleus. Okay, a nucleus was formed around the DNA to help protect it. So those cells that had that structure were more successful. We have these little tiny cyanobacteria and heterotrophic bacteria that end up getting eaten, taken in by one of those bigger, because these cells are getting bigger, one of these early eukaryotic cells, and that becomes the mitochondria. And then we also have, and you can see all this membrane in here, this membrane called the endoplasmic reticulum, which has many different jobs inside a eukaryotic cell. So here's that theory called the endosymbiotic theory. Now, with that theory, we look at time. Here is our ancestral prokaryote. You can see the DNA in there, the cytoplasm, which is the region around the DNA, and the cell membrane or plasma membrane. We start getting infolding of the membrane. to give us some of those structures, that endoplasmic reticulum, our nucleus. So now our DNA is getting protected. And then these guys right here are causing a problem. These photosynthetic prokaryotes, and remember, they're kicking out oxygen. Remember how I said the oxygen it's nasty, it's greedy, it doesn't play well with other atoms, molecules, or even your body. That's what forms free radicals and has to do with aging. So oxygen is just, it's corrosive. I know we need it to live, but it is not a very nice atom. In this case, many, I say many, anaerobic bacteria cannot deal with oxygen. It's one of the reasons you put hydrogen peroxide on your wounds, is to force oxygen in there to kill some of those anaerobic bacteria. Right? So oxygen is toxic to themselves. Well, this guy here, this early prokaryotic cell developed a way to deal with oxygen. And not only did it deal with oxygen, but it also kicked out a lot of ATP. It took sugar, it took oxygen, and was able to kick out a good amount of ATP. We do not know if this big molecule just absorbed it or took it in to eat it, or was the little guy a parasite? Don't know, but they worked well together. The big cell could now cope with oxygen. The little one got protection and food because the big guy fed it. And then the little guy gave the big guy energy, a lot of energy. So this is why we call the endosymbiotic theory. Endo means into, sym means together, bio, life. So these are working together here. They form a great relationship or partnership, and they're much more successful together. Now, somewhere down the line, this happened again. So it happened a second time. All right. So again, we have this group that's going to lead to eukaryotic cells that are heterotrophic, that consume other organisms or nutrients. When this happened again with the cyanobacteria, right, that was the autotrophic prokaryotes, this led to our photosynthetic eukaryotes, like plants. So I always talk about, we're gonna go through all these mass extinctions over Earth's history, but nobody talks about this one. And I feel like it should be mentioned. So as those oxygen levels increased, the anaerobic bacteria that couldn't handle it died. So many bacterial cells died during this increase in oxygenation. The ones that had mutations that allowed them to be successful survived. Some of them that could find habitats void of oxygen survived. But this was a time period where this is probably officially our first mass extinction here. So I kind of have these divided up into anaerobes. Anaerobes means without oxygen, right? No oxygen, uses oxygen. You can see how the anaerobes, they make lactic acid. That's a byproduct of their metabolism, or they make ethanol. Aerobes, they are going to use what's called the citric acid cycle and oxidative phosphorylation. Don't know how much detail I'm going to get into that with you guys. But just know, basically, we're going to take our sugar, C6, H12O6, and break it all the way down to CO2, which is what you exhale. That is our waste product. Both of them are going to make ATP. They both make carbon dioxide, and they both use glycolysis. Okay. So that kind of gives you the first part of early life on our planet, and how we're going to end up moving to different types of varieties of organisms as we go through this lesson with each of our different parts. I hope this was helpful, and I will see you in the next video.